Brown spot or sesame leaf spot or Helminthosporios Helminthosporium oryzixin, Drexra oryzi. Sexual stage, Cochleobolus meabenus. In India, this disease is the principal cause of Bengal famine of 1942-43. The first report of the disease in India was made by Sundararaman from Madras in 1919 and now is reported from all of the rice-growing states. Under highly favorable conditions, the disease causes a reduction in yield ranging up to 90%. Symptoms: the fungus attacks the crop from seedling in nursery to milk stage in main field. Symptoms appear as lesions, spots, on the coleoptile, leaf blade, leaf sheath, and glumes, being most prominent on the leaf blade and glumes. The disease appears first as minute brown dots, later becoming cylindrical or oval to circular. The several spots coalesce and the leaf dries up. The seedlings die and affected nurseries can often be recognized from a distance by their brownish scorched appearance. Dark brown or black spots also appear on glumes which contain large number of conidiophores and conidia of the fungus. It causes failure of seed germination, seedling mortality, and reduces the grain quality and weight. The disease is associated with a physiological disorder known as akioki in Japan. Abnormal soil conditions, deficiency of potassium predispose the plants to heavy infection. Oval to circular spots, olive green to golden brown conidia. Etiology: H. oryzi produces grayish brown to dark brown septate mycelium. Conidiophores may arise singly or in small groups. They are straight, sometimes geniculate, pale to brown in color. Conidia are usually curved with a bulge in the center and tapering towards the ends, occasionally almost straight, pale olive green to golden brown color, and are 6 to 14 septate. The perfect stage of the fungus is C. meabenus. It produces perithecia with SCI containing 6 to 15 septate, filamentous or long cylindrical, hyaline to pale olive green ascospores. It produces terpenoid phytotoxins called ophiobolin A, or cochleobolin A, ophiobolin B, or cochleobolin B, and ophiobolin I. Ophiobolin A is most toxic. These result in the breakdown of the protein fragment of cell wall, resulting in partial disruption of integrity of cell. Disease cycle: the fungus overwinters mainly in the infected plant parts. It is not soil-borne. The infected seeds are the most common source of primary infection. Diseased seeds: 5. Externally seed-borne may give rise to the seedling blight, the first phase of the disease. The young seedlings show infection symptoms soon after germination. Pale yellowish-brown spots appear on the coleoptiles, spreading to cover the other tissues of the seedling. The fungus reproduces on the spots and is disseminated by air currents. The fungus also survives on collateral hosts like Digitaria sanguinalis, Leersia hexandra, Echinochloroacernum, Penicetum typhoides, Ceteria italica, and Cynodon dactylon. The symptoms of potassium deficiency are somewhat similar to that of brown spot, making it often difficult to ascribe the symptoms to fungus attack or nutrient deficiency. Favorable conditions: temperature of 25 to 300 C with relative humidity above 80% are highly favorable. Excess of nitrogen aggravates the disease incidence. Management: 3/4 use disease-free seeds. 3/4 field sanitation, removal of collateral hosts and infected debris in the field. 3/4 crop rotation, 3/4 adjustment of planting time, 3/4 proper fertilization, 3/4 use of slow-release nitrogenous fertilizer is advisable. 3/4 good water management, 3/4 use of soil amendments, 3/4 grow disease-tolerant varieties viz. Baller, 10 Bosnia and Herzegovina convertible marks, R20, Jaya, Ratna, Telehamsa and Kakatia. 3/4 treat the seeds with thiram or kaptan at 4 grams per kilogram and with mancozeb at 0.3%. 6. 3/4 spray the crop in the main field twice with mancozeb at 0.2%, once after flowering and second spray at milky stage.